The overall goal of this permeable membrane insert-based infection system is to study the effects of secreted bacteria toxins on host cells during infection.
This method can help answer key questions in the field of host pathogen interactions, such as how specific secreted bacterial components contribute to host cell responses during bacterial infection. The main advantage of this technique is that it allows for the study of secreted bacterial components that more closely models a physiologically relevant host pathogen environment in the context of human infection. Demonstrating the procedure will be Rebecca Flaherty, a senior graduate student from my laboratory.
To prepare isogenic wild-type Group A Streptococcus, or GAS, and GAS M1T1 5448 mutant strains, start overnight liquid cultures by inoculating 5 to 10 milliliters of Todd Hewitt broth and incubating at 37 degrees Celsius for 16 hours. Centrifuge the overnight bacterial cultures and use the original volume of fresh bacterial medium to resuspend the pellet. Then, normalize the bacterial cultures to equal starting concentrations by diluting the resuspended cultures in additional medium to arrive at the same OD 600. To collect lysates for carrying out signaling analysis and ATP determination assays, plate Hackett cells in 6-well dishes at a seeding density.